Hello everyone, my name is Elton Rodrigues and I am a postdoctoral researcher at IMA UFRJ. Today we will be discussing various aspects of bibliometric analysis, food science, and time-domain NMR. But let us take the necessary first steps and conceptualize the topics at hand. What is food science? And what is the importance of research in this field? Well, food science is an interdisciplinary discipline that focuses on understanding the processes involved in the production, storage, distribution, and consumption of food. It is a critical area because it is directly related to the quality and safety of the food we consume, as well as our understanding of the nutritional and sensory aspects of food. Research in food science covers a wide range of topics, including food microbiology, food chemistry, food technology, and more. It is essential for innovation in the food industry and for ensuring that food meets safety and quality standards. But how do we ensure that the aforementioned quality and safety standards were met? We employ analytical methods, which are essential for assessing the quality, safety, and composition of food products. They provide insights into various aspects from chemical composition to microbiological safety. Some analytical methods are listed below and include spectroscopy methods, for example, UV-Vis spectroscopy, Fourier transform infrared, and nuclear magnetic resonance, which are used to analyze the chemical composition of food, identifying components such as proteins, lipids, and sugars. Chromatography techniques such as HPLC and GC, which separate and quantify compounds in complex food matrices. They are vital for detecting additives, contaminants, and flavor compounds. Microbiological methods, for instance, assess the presence of microorganisms in food, ensuring its safety. Techniques like PCR and plate counts are commonly employed. Texture analysis measures mechanical properties of food, helping to assess factors like crispness, hardness, and viscosity. Instruments like hyometers and texture analyzers are used. Sensory evaluation involves human sensory panels to assess food quality, taste, aroma, and appearance. It provides valuable insights into consumer preferences. Thermoanalysis methods such as DSC and TGA study the thermal properties of food, helping to understand phase transitions and stability. Mass spectroscopy or spectrometry identifies and quantifies compounds based on their mass to charge ratio, and it's a valuable tool for detecting volatile compounds and contaminants. These analytical methods collectively ensure food safety, quality, and compliance with regulations. They are indispensable in research, product development, and quality control in the food industry. Well, in food science, a combination of these analytical methods provide a comprehensive understanding of food composition, safety, and sensory attributes, contributing to the production of high quality and safe products. Among the various spectroscopy techniques available to the food scientists, we highlight nuclear magnetic resonance. Nuclear magnetic resonance spectroscopy is a powerful analytical technique with diverse applications across various academic disciplines, including chemistry, physics, biology, food science, and material sciences. At its core, NMR relies on the interaction between atomic nuclei possessing a magnetic moment and an external magnetic field. When exposed to radio frequency pulses, these nuclei resonate at its specific frequencies, revealing valuable information about the sample structure, dynamics, and chemical environment. Some applications of NMR in food science are given. A fundamental application of NMR in food products is the precise determination of water content. This is critical for ensuring the stability and quality of food products. NMR is available in lipid analysis, allowing the identification of different types of fat and oils present in food, including unsaturated and saturated fatty acids. The technique is widely used in texture analysis, enabling the assessment of firmness, viscosity, and other sensory properties of foods. NMR is also essential in identifying bioactive components such as antioxidants and phenolic compounds, contributing to our understanding of the health benefits of foods. NMR plays a fundamental role in quality control, helping to detect adulterations, contaminants, and ensuring the authenticity of food products. In the development of new products, NMR assists in formulation, optimization, and process monitoring, saving time and researches. NMR analysis is valuable in flavor and aroma research, allowing the tracking of volatile compounds, contributing to the sensory characteristics of foods. NMR may be divided into two broad categories: frequency domain NMR and time domain NMR. Time-Domain NMR, TD-NMR, is a powerful analytical technique used in various fields, including chemistry, physics, and notably in the study of materials, especially in the characterization of complex systems and soft matter. TD-NMR relies on the interaction between atomic nuclei and magnetic fields. By subjecting a sample to a strong magnetic field and applying radio frequency pulses, it measures the time-dependent response of nuclear spins, providing insights into the sample's properties and molecular dynamics. TD-NMR finds applications in diverse areas such as polymer science, food analysis, and medical diagnostics. It is valuable for studying molecular mobility, particle size distribution, and phase transitions in materials. Notable advantages of TD-NMR include non-destructive testing, minimal sample preparation, and its ability to provide quantitative information about various sample characteristics. TD-NMR continues to advance research across multiple disciplines, enabling scientists to gain deeper insights into the structure and behavior of materials, contributing to innovation and problem solving in various industries. But how is time domain nuclear magnetic resonance used in research in food science? Well, TD-NMR is a technique used in food science to study the composition and properties of foods. It allows for non-destructive analysis of food samples, which is desirable, providing detailed information about molecular structure, the dynamics of water molecules, texture, and other parameters. It is often used to assess the quality of food, such as determining water content, lipid profiles, and protein structure in food products. Additionally, TD-NMR can be used to monitor changes over time, which is crucial for ensuring the stability and quality of food products during storage and transportation. Well, this is all well and good, but how can one navigate in an informed, efficient, and reliable manner across the various domains of TD-NMR applied to food science? Is there a way to categorize publications in this field, comprehend their correlations, and forecast trends based on extensive data volumes? Well, there is a way of doing exactly that. We're talking about bibliometric analysis. Bibliometric analysis is a quantitative research method used to assess and analyze scientific literature, academic publications, and research trends. It involves the examination of citations, publication patterns, collaboration networks, and the impact of research within a particular field or discipline. The primary aim is to gain insights into the evolution of a research area, identifying influential authors and institutions, and track the dissemination of knowledge. Bibliometric analysis is widely used in academia, research institutions, and industries to inform strategic decision-making, assess research impact, and identify emerging areas of study. Some key steps in bibliometric analysis are as follows. Database search by conducting comprehensive searches in reputable databases like Scopus, Web of Science, and PubMed using relevant keywords such as TD-NMR, food science, and related terms in our case. Collecting data on publications, including titles, authors, affiliations, keywords, publication dates, and citation counts. The correlation analysis utilizes is utilized to identify relationships between publication variables such as author collaboration networks, keywords occurrence, and citation patterns. Then we proceed to cluster analysis. We apply cluster analysis to group publications with similar themes or research topics. This aids in understanding the subfields within, for instance, TD-NMR applications in food sciences. The trend estimation is a step which one employs trend estimation techniques to identify emerging research areas, the growth of specific topics, and potential research gaps. And finally, the visualizations, where we create visual representations such as co-authorships networks, citation maps, and keyword clouds to enhance the understanding of the research dynamics. Bibliometric analysis provides several advantages, including the ability to quantitatively assess research impact, identify influential authors, institutions, and journals, and monitor the evolution of the field. And finally, developing a well-informed research strategy based on bibliometric insights can guide your explorations of the topics at hand. For instance, TD-NMR applications in food science, helping you stay current and contribute meaningfully to the field. But how does bibliometric research contribute to the advancement of food science and time-domain NMR? Well, bibliometric research plays a vital role in both food science and the field of TD-NMR by enabling scientists to assess and understand the evolution of these fields over time. Through bibliometric analysis, researchers can identify research trends, academic collaborations, key authors, and prominent institutions, as well as map the dissemination of scientific knowledge. This aids in identifying research gaps, efficiently allocating resources, and promoting scientific progress. Furthermore, bibliometric research can be employed to pinpoint emerging research areas and influence the future development strategies in both food science and TD-NMR. Navigating the diverse applications of TD-NMR in food science requires an informed, efficient, and reliable approach. Understanding correlations, estimating trends, and classifying publications in these fields based on extensive data volumes is essential for meaningful research. By employing bibliometric tools and techniques, researchers can systematically analyze and interpret large volumes of scientific publications related to TD-NMR in food science. Well, with all that being said, here we have some of our results. We conducted a search on the Scopus platform, utilizing the search tool for article titles, abstracts, and keywords with keywords food science or food chemistry or food and relaxometry or NMR. Subsequently, a file containing all the located articles was exported, and this file includes information about authors, document titles, publication years, citation counts, DOIs, abstracts, and author keywords. Additionally, on the Scopus platform, another tool was employed. From the articles found in the keyword search, the search result analysis tool was utilized, enabling the exporting of data regarding the number of articles published per year. With this data, it became possible to generate graphs such as the one displayed right now, exhibiting the variation in the quantity of publication over the years and maps depicting connections and density among the subjects addressed in the articles using the VOS Viewer software. Our results revealed a rapid growth in publications on the subject over the years, indicating that this indeed is a topic that is increasingly capturing the interest of the scientific community. Well, the data from the connection and density maps reveal that TD-NMR is intrinsically linked to various food topics such as antioxidants, polysaccharides, seeds, fruits, water, oils, and so forth. This demonstrates how the TD-NMR method can permeate research on food topics from various angles. Many other terms related to NMR also appear as relevant clusters: NMR spectroscopy, NMR imaging, etc. Quality control, food storage, and food processing emerge as clusters intrinsically linked to NMR techniques as well. Uh, in another node, marked in blue, variations of words related to the bibliometric space of NMR and TD-NMR are also present. Here, we highlight the possibility that the relevant literature may not rigorously distinguish between terms like NMR and TD-NMR, which can hinder research on the correlating or the correlation of those spaces. So the hypothesis of misrepresentation of the terms used in the assessed papers is reinforced by the analysis of the density map obtained from the conducted search. Here we clearly observe that the more recently used terms, represented in warmer colors, are concentrated around NMR, NMR spectroscopy, and proton-nuclear magnetic resonance. The data reveal the importance and continued relevance of TD-NMR and related spectroscopy techniques in food science, a trend that shows no sign of weakening in the near future. The immediate conclusions are that relaxometry or TD-NMR is a valuable tool in food science with applications in various areas. The increase in the number of publications indicates a growing interest in this field. Furthermore, TD-NMR's ability to relate to various aspects of food reinforce its relevance and versatility in food research. In summary, navigating the multidisciplinary landscape of TD-NMR in food science is made more manageable and insightful through the systematic applications of bibliometric analysis. It equips researchers with the tools needed to classify publications, understand correlations, and estimate trends, ultimately facilitating informed and efficient research. Well, that's all we had for today's presentation. Thank you all for your interest and time. I may be reached through the email displayed on the screen. See you next time.